One common misconception about gene structure, which comes largely from the central dogma of molecular biology, is that a gene in DNA is limited to the region that gets transcribed into RNA. Even if we expand the central dogma to include the transcription of RNAs that do not get translated into proteins, i.e. the non-coding RNAs, it is still easy to think that genes are only those regions of DNA that get transcribed into RNA. But this is only partly true. It is true that genes contain a coding region, which is the part that gets transcribed into RNA. This coding region begins at what we call the transcription start site, or TSS, also known as the +1. This corresponds to the first nucleotide that gets transcribed and therefore to the first nucleotide of the RNA molecule. But genes also include additional segments with distinct functions. For example, in order for RNA polymerase to know where to begin transcription, there must be DNA sequences that signal "start here." These are called promoter sequences, and they are found immediately upstream on the 5' side of the coding region. Promoters control the access of RNA polymerase to the coding sequence and ensure that transcription begins at the correct location and in the correct orientation. Genes also need information to signal where transcription should end. In bacteria, this is achieved by termination signals, DNA sequences downstream of the coding region that instruct RNA polymerase to stop. In eukaryotes, the process works differently. For protein-coding genes, RNA polymerase keeps transcribing beyond the coding region, but the cell uses only a shortened version of the transcript. This is due to polyadenylation signals, or poly-A signals, which direct cellular machinery to cut and process the RNA at a specific site near the 3' end. So while bacteria terminate transcription directly after the coding region, eukaryotes extend transcription further and then actively process the RNA to create the mature transcript. As we'll see in class, this mechanism applies to messenger RNAs, but not to all eukaryotic RNAs. Now, let's return to promoter sequences. The sequences that direct RNA polymerase to the TSS are referred to as core promoters. These simply specify where polymerase binds and in which direction transcription begins. In addition, there are other sequences in DNA called regulatory sequences, which together form what are sometimes called regulatory promoters. These work indirectly. Proteins bind to these regulatory sequences, and through that binding, they influence whether RNA polymerase can access the TSS and how much. In this way, they can either facilitate or inhibit transcription. Regulatory sequences can be located immediately adjacent to the core promoter, and for a long time, scientists referred to the entire region as simply the promoter. Even today, you'll sometimes see the term promoter used to mean both the core promoter and nearby regulatory sequences. But in eukaryotes, it gets a little more complex. Regulatory sequences don't have to be right next to the gene they control. They can be located far away, even on different DNA molecules. When regulatory sequences act at a distance, we call them enhancers. Historically, these were discovered as elements that increased gene expression, and hence the name. But today we know that distant regulatory sequences can also act as silencers, which repress transcription, or as insulators, which create boundaries that prevent the influence of other regulatory elements onto a given gene. The main point here is that genes are more than just the coding regions that get transcribed into RNA. A gene also includes promoter sequences that direct and regulate transcription, termination regions that signal when transcription should stop or where transcripts should be processed, and in eukaryotes, sometimes even long-distance regulatory elements like enhancers, silencers, or insulators. Okay, I hope this overview helps clarify the broader structure of genes, and I'll see you in the next video.